Well, welcome everyone to this fireside chat between Allan and myself. I don't need to make an introduction because I think most of you know I'm Bob Smith, chairman of the board here at ARM and venture partner with OrbiMed. Allan, why don't you take a few minutes to introduce yourself?
Thanks, Bob. Yeah, Allan Reine. I'm the Chief Executive Officer of Prime Medicine. We are a gene editing company focused on technology called Prime Editing. I've been with the company since January, so I'll say I've been in the cell and gene therapy space since January of 24, so cell and gene therapy space for just over a year and a half. And it's pretty incredible. This is my first time coming to this conference, and really incredible to see this community and kind of what you've built here. And feel very fortunate and really excited to be part of it.
Great. So do you want to kick off and maybe give us an overview of Prime Medicine and a little bit of the story of the company and your role there?
Yeah, I would love to. Prime is, again, a company really focused on addressing disease. And to do that, both genetic diseases, but also non-genetic diseases that have a gene editing approach to treat them as well. So we're really looking at how we get to safe, effective, and hopefully curative treatments, which can really offer lifelong benefit to patients. And that's really what this is all about for us. To give you a little history of the company, the technology is only about 5 to 6 years old. So this was developed in David Liu's lab in 2019. David Liu, also very well known for base editing, a company called Beam, which also came out of David Liu's lab. Him and someone named Andrew Anzalone, who works at Prime, is one of our scientific leaders. Him and David came up with the technology. And then as I think about the company kind of formation in the first few years of the company, it was really trying to figure out where we should take the technology into what types of diseases and what sort of tissue types. So I always think about gene editing companies, and I'm sure people in the audience can understand this well, is they're really two different companies within one, right? There's a gene editing technology that we all have. Sometimes there's more than one gene editing technology within a company, but then we're all delivery companies as well, right? So it's not just can you edit the genes, edit the genome, but can you actually deliver your cargo in a safe and effective way to the cell type that you're trying to get at. And so we really built that over the first few years and really got to proof of concept, showing that we could deliver to the liver safely and effectively. We could obviously do this ex vivo to hematopoietic stem cells and other cell types. We've shown we can deliver safely and effectively to the brain. Again, all in preclinical studies, but just showing sort of the power of the technology. As I think about sort of where we are now, we signed our big first strategic collaboration with BMS last year for ex vivo CAR T cells. We've now shown our first clinical data, and I'll show you that in a minute, but really demonstrating for the first time we think we have essentially a functional cure for 2 patients in a disease called chronic granulomatous disease. And we've really focused the company now, in the near term at least, on our 2 liver programs and cystic fibrosis that I know we'll talk more about. And then as we think about the future, it's really how do we get Prime Editing to the most number of patients throughout the world, as we think about how powerful this technology is, the difference we think we can make in patients' lives, and really thinking about how we can kind of get to more proof of concept and ultimately to commercialization over the next few years. So to tell you a little bit about what the technology is, so we think about this as the most versatile gene editing technology. Well, why do I say that? So if you think about sort of CRISPR-Cas9, you're making a double-stranded break. They do the sort of that search part within the genome really, really well. So how do I find a very specific place within the genome where I want to make my edit or my cut? And that's the incredible, obviously now Nobel-winning technology that they came up with. But there are some limitations there. You're making a double-stranded break. You have some unwanted off-target editing and other things that can happen. And in addition to that, it's very good at knocking things out, but how do you make changes? How do you put things back in? How do you correct many different types of mutations? Then obviously base editing came along where you can make kind of one change, one base pair to another, in 4 instances. And then by adding reverse transcriptase to the mix with a modified Cas enzyme, so we're making a single-stranded break. And then with a reverse transcriptase, we're putting a RNA template into our guide. So now you can transcribe base pairs directly into the genome. So it's not just knocking something out, you're making a single-strand break, and then you are correcting the genome to whatever, likely back to wild type, because that's usually the goal. So we can correct deletions, insertions, what we call point mutations, so not just transitions, but what are called transversion mutations. We do something called hotspot editing. We do this in cystic fibrosis as an example, where by correcting a longer stretch of DNA, I might be able to correct a mutation that I have and a mutation that you have, both separate mutations, but with one gene editor. So it's the first sort of technology that can do that. We can go after what are called repeat excisions, some of those diseases like Friedreich's ataxia. And with our passage technology, we can do almost full gene insertion. So with a very specific, what we call landing pad, insert a very long stretch of DNA. This just highlights that we really don't see any off-target editing with our technology. We don't see any translocations, chromosomal rearrangement. So it's a really just powerful technology in terms of what we can do in terms of the editing, but also the safety as you think about sort of the long-term safety when you're editing the genes. And the last slide I'll show you or I guess I have one slide after this showing our pipeline, but this is an important slide showing our first clinical data. So I won't go into too much detail here, but just this is a disease called chronic granulomatous disease. You essentially want to get, this is an ex vivo HSC therapy. And the goal here is to get high efficiency editing. You want to know that these patients have at least 20% of their HSCs edited. That should be sort of a curative level for these patients. We know that from looking at allogeneic transplant. We are well above those thresholds as we look at the 2 patients that we treated. These patients also have pretty profound inflammatory-like disease, and we've seen incredible reductions in real hard clinical endpoints as well. So we're very excited about this data. Unfortunately, that program, there's only about 40 patients to treat in the US, so we had to make the very difficult decision not to move that forward. There's a New York Times article that just came out on this that I encourage you to read. We are trying to figure out ways and have discussions with the FDA to figure out if there's a way to get this to patients and maybe even to commercialization potentially on the current data set. And as you look at our pipeline today, we're focused on our 2 liver programs, both for Wilson's disease and alpha-1. We have INDs for both programs next year. And then for cystic fibrosis, where we continue to make excellent progress, and with BMS, our CAR-T collaboration.
That's great. I mean, incredibly versatile, cutting-edge technology. I mean, I'm really interested to hear a little bit more about how you're thinking about applying this in the cystic fibrosis space, and maybe describe a little bit about some of the technical challenges with cystic fibrosis and how you can see this versatile technology platform addressing the disease.
Yeah, so as you think about cystic fibrosis as a disease, and what Vertex has done to really change the course of that disease for patients, like this is what it's all about, right? Like these are the stories to me that just, that really make you proud to be part of this sector. Like it's made an incredible difference to these patients' lives. Like this is a disease where, patients could live into their teens or 20s and often not beyond that, and had really debilitating disease even throughout that period. Along comes, Trikafta and the development of a medicine where they can lead fairly normal lives now. Like that's incredible. There are, though, about 10 to 15% of patients that have mutations, what are called sort of nonsense mutations, that are not responsive to that therapy. And so the Cystic Fibrosis Foundation is very focused on how do we get that 10 to 15% of patients, how do we ensure that they can be treated in the same way and have the same outcomes to those that other group of patients? So they're very fortunate to have a lot of money to work with from the royalty that they sold on their Vertex drugs, and they're using that to really try and figure out how to now push those sort of other mutations forward. So we are working with them, and they are largely funding the Cystic Fibrosis Foundation, largely funding the program that we have today to try and address those. So how are we doing it? So we have two approaches. So if you saw that slide that I showed before, we have our passage approach. So that's doing like a larger insertion. Where we can treat many mutations with one approach. It's a more complicated approach for sure, because you need a DNA donor when you're doing a large insertion. So, which means it's not as easy to get that delivery right in vivo. But the second way we're doing it is, and probably, again, I'm not saying anything's easy because it's actually all really challenging, and I'll get into what the challenges are, but making those specific, just correcting those very specific nonsense mutations. And we've made a lot of progress, and we've put out some data showing we can get very high editing efficiency in the right cell types, in the right medium, which is important when you're looking at CF, because it's not just can I get editing, but can I do it in a certain culture that shows you can do it sort of in what a patient's lung airway looks like. And I'd say the second piece is, can we deliver it in vivo? And we've now got good data to show that we can deliver it in vivo, but we're still working to, I'd say, optimize that. We have two different approaches there. So we have both an LNP approach and we've got an AAV approach, and I think we're making good progress with both. I think one of the challenges when you're thinking about cystic fibrosis is on the delivery side is getting to the right cell type. So you can get to the more superficial, call it epithelial cells, which is great, but if you treat only those will turn over, over a period of time, that cell type will turn over, and you would lose efficacy if you're not hitting those true, bronchial epithelial progenitor cells. So we have a lot of assays to understand, are we hitting those cell types? And that's what our goal is here. And so when we think about if we're successful in vivo, to us that means, are we hitting those bronchial epithelial progenitor type cells? And there's a lot of different animal models and different animals that you could work with here to try and get to data that really demonstrates what we think will translate in humans. So although there are challenges, we continue to make great progress, and I'm hopeful in the future we'll have a medicine that can really change the lives of these patients.
Yeah. And could you just share a little bit about the nature of the relationship with the CF Foundation? When I was at Pfizer, we actually had a multi-year collaboration with them, so I have a little bit of a sense about how they work, but maybe for the benefit of the audience, I think many look at the CF Foundation as like best in class in terms of patient advocacy group, philanthropic funding of research, but just share a little bit about the relationship and how you have been working with them.
Yeah, I mean, they are, and I'm not just saying this because they give us a lot of money. They're an incredible group to work with. They've hired really intelligent people who know the science really, really well, both on the delivery side, but also whether it's the gene editing side, mRNA, whatever technology they want to look like. They know every single CF modality technology that is out there. They obviously have seen a lot, some under CDA, some not, so they can only share certain things. But they have a very good idea of what they think works, what they think will work, what won't work, and they're extremely collaborative. And I know they're very collaborative with us. I also know some of their other partners, and I know how collaborative they are with them. And they really have a mission. They are really trying to solve this very difficult problem. And I was just we had a JSC recently that I attended part of, and it's just incredible the engagement that you get from them. And it's I'm sure we've all sat in certain JSCs throughout our careers, but this one just has a great feel. Like we're really just both working together to try and figure out what are the best ways to solve these very challenging problems in front of us.
Yeah, our experience with them was very similar. I mean, their level of scientific expertise, the relationships and the network that they have. Maybe just one kind of like futuristic type question is, you're doing this early research collaboration with them. What's kind of like the end goal in this? Are they also going to be involved in participating in the clinical trials, or do you view this as kind of a limited research-only funding and then you're going to run with it thereafter?
Yeah, I mean, they've been involved in funding clinical trials in the past. So I wouldn't say definitively today that that's where we'll go, but I could definitely see a likely scenario where there is continued collaboration even into clinical development. And ultimately, I think, or at least what I've heard, is they can be very helpful once you're in the clinic, right? Yeah. They've got a fairly good idea on sort of patients and where patients are located, how to ensure you're enrolling the right patients. So at least from the conversations I've had, the ability to have them on board, whether they're funding it or not, but just being a partner to help you in the clinic, I think is an important thing.
Yeah, and I think they're great. Maybe just going back to the technology platform. I know you look at the whole field of genetic medicine, you're a roughly now 5-6-year-old technology platform. What do you see as, and I know it's quite versatile, but what do you see as kind of the relative trade-offs of its benefits versus its challenges? And where do you see like the future application of prime editing versus base editing, gene editing?
Yeah, it's a great question. And as I think about it, I think we're just scratching the surface now for these technologies, right? I think it's very easy, and obviously we saw a ton of excitement as we looked 5 years ago, or kind of where the market was and the funding was and everything else. And there was probably a little bit too much hysteria at that point in time. But I think and then kind of market cycle, and we've kind of, I think, really seen the bottom. And now it's sort of like I think people are viewing this in a little bit of a different way. Now, why is that? Well, one, you have a technology that was very early, right? So you had very early clinical trials, and you didn't have a lot of data to go on, right? I think now we've sort of built up within gene editing specifically, a critical mass of data that we kind of really can understand, both from a safety and efficacy standpoint, not everywhere, but definitely in the liver, and maybe some other tissue types, and definitely ex vivo, kind of, that we can do this safely, we can do it with high efficiency, and we can do it, efficaciously. That's really important. And so I think as we think out into the future, I think it's going to be about what other tissue types can we deliver to. And to me, it's not a question of if, it's a question of when. I think if you look 2 years ago and you said, how are we ever going to deliver to the brain? And we're all trying to figure out the systemic AAV delivery vehicles that'll get us there, right? And it's a challenge, right? It's a big challenge. And I can tell you right now, preclinically, that was a challenge. But now, and no one would have ever thought like, oh, we're just going to go direct injection and that's going to be the answer and that's going to be a way we can do this. But now we've seen this with both, Neurogene, we've seen a different type with Taysha. We've seen what Unicure has just put out, which is, I think, just incredible data and a really novel way to deliver a drug. So you're just seeing, you're seeing delivery, I think, improve every day. And again, I think we're going to get to a point where we're going to be much better either with AAV or non-AAV type approaches to getting to different tissue types. And once we can unlock that, it's almost like the opportunities are limitless. Yeah. But you talked about the challenges, and that is still a challenge, right? Yeah. So the opportunities there, but we still need, please fund companies that are working on these delivery breakthroughs. We know we're not going to solve, many of those internally. Like we're going to focus on certain things. Like we will solve some of those, but the more capital that can get at really solving some of these difficult problems really opens up, I think, the ability to ultimately treat millions of patients across a number of different disease areas.
Yeah. You kind of mentioned delivery as a challenge, and I'll just give a little foreshadowing to those of you who will be here Wednesday morning. I also work as an advisor with the Focus Ultrasound Foundation, which is a completely different part of medicine, but there's kind of a convergence of outside of the traditional realm of viral mediated or LNP mediated genetic medicine and how to enhance delivery. The field of focus ultrasound to improve opening of the blood-brain barrier has now been demonstrated in both preclinical to be quite exciting, and they're also doing a lot of work with also large organs, an example, the heart. So Fred Padilla, who is one of the scientific directors for the program there, will be presenting on Wednesday. I encourage all of you, if you're not aware of it and you haven't heard about it, listen in on Fred's presentation. It is really a remarkable advancement in the convergence of technologies that you wouldn't have other thought would make sense for enhancing genetic medicine delivery. But these are the types of exciting things that I think the field is kind of stimulating from fields that are adjacent or probably quite distant from what you might think of as traditional delivery technologies. So that's a little bit of a plug for them, but just tell us a little bit more about where you see the kind of next wave of pipeline programs. You got their CF program, you have your liver directed. Are you going to continue to concentrate on those particular tissues, or are you thinking about building out a broader pipeline? And I know you've gone through a portfolio rationalization. Yes. Maybe just share a little bit for those who are similarly situated. You can't fund everything, so how do you decide which ones you will fund?
Yeah, so maybe I'll start with that and just say, when I joined the company in January 24, we had I think we had 18 programs listed. We probably had more that we were working on, which we know is not sustainable. I don't think we ever thought we were going to take 18 programs, through to the clinic on our own. But there is something to be said for focus, right? You can only solve a certain number of problems, at one time, and you can lose some of that focus if you go kind of too diffuse. And we were doing AV, ex vivo through electroporation, other ways. We're doing LNP delivery. We're doing a number of different tissues, and everything has its sort of like its own challenges. So I think and then it was really understanding. Because we're businesses at the end of the day, we have to really understand where are the real sort of commercial opportunities. So my first 3-4 months of the company was really just trying to put together a strategic framework to say, what should we focus on? How can we build near-term value? And the more near-term value we can build as a company, the bigger we can kind of, the more prime editors we can make over time. That's sort of how I think about it, right? The more success we have early, the more we can do for patients in the long run. So let's not, if we focus on 1 or 2 very small disease areas, that may be profitable, may not be, that's not going to get us there. So what's the right strategic vision for the company? And so that really got us to, as you said, really focus on the 2 liver programs, obviously our collaboration with BMS and then CF. So I think as we think about CF, we want to get to not just those 10 to 15% of patients that I talked about, but through the different hotspots we'd go after. We can hit Del 508. We can hit, we can potentially treat 90+% of CF patients. So we're not going to just sort of stop with that, few different mutations that they're trying to treat. I think when it comes to liver disease, the great thing there is this is a modular platform in a sense as we think about the liver. We've got an LNP that we think will be, safe and effective. I think with that, I can tell you our Alpha-1 program is much cheaper and faster to the clinic if we hit our timelines than our Wilson's disease program, because we can hopefully lean on a lot of the learnings in terms of obviously CMC, but also some of the tox studies and other things that we hope to not have to repeat. And so if you think about layering on additional liver indications, and there's some interesting ones to go after that I think are really prime for prime editing, that to us is a really sort of cost-effective way to bring more programs into the clinic. So I think additional liver programs is an important part of the strategy. I think beyond that, I mean, if we go back to neuro that we talked about, there are just hundreds of different diseases to ultimately go after in the brain. We've shown some really strong proof of concept in some of our preclinical models. Again, I don't think it's a capital allocation that we're going to kind of go after today on our own, but it's something that I hope we do with partners, because ultimately, I think if we're successful there, there's just a tremendous amount of opportunity. I think about cell therapy as another place, especially from a partnering perspective. I think what you can do, whether it's iPSCs or other using prime editing versus other gene editing approaches, we think this can be a sort of best-in-class way to go about cell therapy. Again, whether it's CAR T, whether it's iPSCs and whatever cell type you're trying to make. So we think there are a number of different areas that we'll continue to explore. And then some of the programs that we discontinued, like our retinitis pigmentosa program or some of our hearing loss programs, we actually have incredible data. So the ability to figure out how to actually get those to patients one day, I think is important, for me as well, just thinking about the company and what we can do for these patients if we're successful.
Yeah. I mean, it seems like in some of those indications, as you build out multiple liver indications, and even within a specific disease like CF, where you might have different forms of the prime editing approach dependent upon either the insertion or the hotspot corrections. If you think about the regulatory framework, how is the work that ARM and other regulatory agencies are looking at gene editing as a platform? How does that regulatory framework, if it evolves the way that we hope, how could that help advance all of this work that you're doing?
Yeah, I mean, first off, I think ARM's doing, from what I hear, just incredible work in Washington and really, is a real advocate, obviously, for the space. But to me, the FDA at least, seems to really get it. So when we don't like to talk about our regulatory interactions in detail, but our hope and belief is, especially as you're thinking about the same disease and multiple mutations, I think the FDA, or I hope the FDA, will be very accommodating in thinking about you're not going to have to repeat everything to take, mutation number 2 into the clinic. Yeah. And hopefully over time, if we show good translation, maybe that'll mean you can go almost right to commercial with very, very minimal data. That's sort of the hope in the future. My hope even beyond that is, can we do multiple editors within the same tissue type, all under one IND. So there's, I don't think the FDA is necessarily there yet, yep. But hopefully with enough experience, we and others can help demonstrate that that's not an unreasonable place to be. Yeah. I do think other countries, regulatory agencies are not quite where the FDA is today. I'm hopeful ARM and others maybe can help there as well, because I think there is a little bit more education as you think about the UK, EMEA, and others in trying to figure out how to advance these therapies. Again, what's critical is, can we do this in a very cost-effective way? I think the FDA understands that. Yeah. Let's do what's necessary. Let's get rid of what's redundant or unnecessary in terms of costs and development. So I'm hopeful that the other regulatory agencies are a little bit behind, but will catch up. But it'll probably take a lot of work by ARM and others in this room to get them there.
Yeah, Mike Lemke and the team are doing a great job, as I fully agree with you. And I think, we had some discussions yesterday at the CEO forum about how we've become much more, I would say, blow up the current constraints in the system. Let's think about ways to do things faster, less expensively, and with higher quality. And I think this whole gene editing or base editing, prime editing as a platform is probably an example where you can make a really big impact. You could limit the amount of investment, reduce all this redundancy and scale, and you could probably scale really quickly. So I view it as a real opportunity for the sector, and probably one of the most exciting things that is kind of evolving in the near term for our sector. We do have a few minutes. I don't know if anyone in the audience has a question for Allan. We're certainly Happy to take any questions. I see one here. You might need to just speak a little bit louder. Do we have a mic?
I can repeat it.
Okay.
You use the two words, my favorite words, constraints and limitations, in the CEO meeting yesterday. And then you talked about the marketplace acceptance. And then the unspoken is how are you going to make some money out of this? So how do you address scientific excellence with markets that need to have something, and you're working on the tools and the therapy to do it so everybody's happy about the scientific success as well as people making money? How are you guys being, I got to give Prime credit. Full disclosure, I'm an investor. Thank you very much. What you've been doing, Allan, I love it. Okay. But is it a function of your visibility on I'm going to go after big markets? I mean, is that the key to trying to take away some of those constraints and limitations where people believe the application is way beyond just, kind of this 100 of that and so on and so forth? That's kind of a long question.
Yeah, no, I'll paraphrase that back. So for some reason, your favorite words are constraints and limitations. And given the constraints and limitations that we've been facing, and obviously you want to go down in terms of scientific excellence, but how do you marry that with sort of what the market expectations are, and how do you ensure your investors are making money, as you as a company can make money, while still sort of pursuing scientific excellence, and have we done that through focusing on larger indications? The answer is, frankly, yes. Like, we have focused on larger indications for that reason. I kind of call it the unfortunate reality, right? Because there's a lot of diseases that we can treat. There's a lot of diseases we can treat that will never become profitable treating. Chronic granulomatous disease is an example of that. It's heartbreaking for us not to take that forward, right? It really is. We're going to try and figure out how to get patients that medicine, but it's heartbreaking for us not to take that forward. But yes, we need to focus on things where, first off, do we think we can have something that's going to be first off, is there an unmet need, right? Is there a place for our medicine? Second, do we think we have a differentiated approach, right? Do we think we are either going to be we don't have to be first, but we have to believe we're going to be best, right? And oftentimes, if we're taking in the case of a mutated protein back to wild type, wild type meaning normal protein, we often think that we can be best. And if we can do that in indications that can be very commercially successful, then we've got a formula that works, right? I think over time, hopefully, there are going to be ways, and frankly, some of this might come from the company, but some of this might come from public-private partnership or other things that are going on now. You've just seen something by ARPA-H get launched called Thrive. Like, they're trying to figure out, the government is really actively trying to figure out how, that sort of no patient left behind. How do we go after these smaller indications? So some of that might not get done by us, but hopefully with those types of initiatives, we can actually contribute and help some of those, whether it's an academic center or someone else, really advance these therapies, for less prevalent indications. I hope I've answered your question, but.
Yeah. Great question, thank you. Anyone else in the audience has a question? We have a mic available if anyone would like to ask a question. We have one in the front here, thank you.
Hi, a very nice talk, Mike Lemke with ARM. Are you concerned about, in cell and gene therapy and gene editing specifically, competitiveness with China? And is that an opportunity or a challenge? How do you look at that?
Yes, and I don't know. So yes, I'm concerned. I'm not concerned about our IP, right? Like, I think it's we feel very confident we own the IP for prime editing, right? Globally, essentially. That doesn't stop companies in China from pursuing, prime editing or base editing or anything else, right? There's plenty of pipelines that look very similar to our pipeline and other companies that I know. So yeah, I mean, it's a concern for sure, right? Are they going to do things? Are other companies going to go and partner? The thing that at least gives me comfort is I know we own the IP. So if they want to take any of those programs and try and bring them into the US or other countries, well, they're going to either have to take a license from us, or they'll be paying us in the future in some way, shape, or form, or they'll be blocked altogether. So, but yes, I mean it absolutely is a concern. That being said, the other side to it is, do we want to do things in China? Clinical trials are a lot cheaper. There's different ways to think about. Can we be more efficient or do things in a different way, kind of using China? And so, we're not the experts there. We're doing a lot of work to try and understand China better. I think it's really important for every company to really think about, yes, what's advancing, but also, are there ways to actually use what's going on in China to our benefit, so that we can sort of piggyback on what they're doing, which is, very cheap, quick ways to get to data.
Yeah, thanks for the question, Mike, and great response, Allan. Anyone else? We have 1 minute left if anyone else has a question for Allan. I don't see anyone raising their hand. All right. Well, Allan, thank you.
Yeah,
I mean,
thank you.
I think it's a really exciting company that you and your team have built with tremendous versatility and potential. I'm really excited to be following your success over the coming years. And thank you for being an ARM member and for spending time with us here and sharing your experience in getting people excited about Prime Medicine.
Thanks for having us here.
Please.